We all know that genetic engineering is a very powerful technology, and we all suspect that as it develops and gets refined and finds greater application in medicine and agriculture, the things we're going to be able to do with genetic engineering will just blow our minds and transform our societies. These are big words, big claims, but the science is incredible, and these big claims can get backed up with empirical research. For today's science news, I want to bring you one of these studies that demonstrates the raw power of genetic engineering. Researchers from the University of Chicago and from Peking University and Guizhou University in China have worked together to create a method that can boost plant growth by a huge margin, improving root size and, most importantly, the yield. The improvements they were able to achieve are really impressive. So first, let me explain a bit of the background, and then I'll get into what they were able to achieve and what it means. To keep this simple, all organisms have a genetic code, and within that genetic code, there are genes, which are small segments of the code that, when they get expressed, can produce proteins, and those proteins can do stuff. Other genes are regulatory genes, which means that they can help regulate how other genes, like those protein-coding genes, get expressed. Some of these regulatory genes can be turned on or off through chemical markers, like methylation or phosphorylation or adenylation. In these processes, some kind of signaling molecule is added to the genetic code, so that when the DNA is read during transcription, that signaling molecule can tell the DNA transcribing enzymes to read or to skip this particular gene. Sometimes the chemicals that transcribe the DNA can also be modified, and this can influence what genes get expressed as well. Now in this instance, we're going to look at the methylation of the chromatin, which keeps it bunched up together and difficult to transcribe. So genes that exist within this bunched up methylated region of the chromatin, they're not accessible to the enzymatic machinery that reads and expresses those genes. In this research, the scientists used a human gene that coded for a protein called FTO. This protein is important because it can demethylate RNA. It can remove a methyl group tag from RNA, and this alters both the structure of the RNA and how it expresses genes. They found that FTO increased the levels of open chromatin and RNA with long polyadenylated tails, which would make large portions of the plant's DNA easier to read and express. They also say that, quote, elevated tissue-specific mRNA species encoding proteins play functional roles in root cell proliferation, tiller formation, and photosynthetic efficiency, contributing to elevating crop yield and biomass, unquote. Here's where things get really crazy. The plants that they altered with the FTO protein saw an approximately 50% increase in root mass, and a subsequent 50% increase in yield in both potato plants and rice plants. In addition, all of the modified plants were more drought resistant, and unbelievably, they were even more efficient photosynthesizers. Photosynthesis has a notoriously low efficiency, due in part to a protein called rubisco, so anything that can increase photosynthesis efficiency, even by a little bit, is amazing. And the fact that they saw improvements in both potatoes and rice is also really important, because these are distantly related plants from each other. The two major clades are eudicots and monocots. This suggests that whatever the FTO protein is doing to the RNA in these plants, it's affecting some kind of conserved system that may be shared across most or maybe even all plants. To quote University of Chicago professor Dr. Xuan He, quote, the change really is dramatic. What's more, it worked with almost every type of plant we tried it with so far, and it's a very simple modification to make, unquote. The researchers believe that this process works through downregulation of the M6A pathway in plants. This M6A pathway, this is a self-regulatory pathway that constrains plant growth. It's an important regulatory factor on plant growth, because if the constraints were totally disabled, plant growth could be unconstrained, and this could lead to potentially harmful tumor-like structures and other weirdness. The presence of the FTO protein in the plant's tissues appears to not disable, but to downregulate this M6A pathway, so that plant growth is upregulated, but not completely unconstrained. Dr. He said, quote, "This is a brand new type of approach, one that could be different from GMO and CRISPR gene editing. This technique allows us to flip a switch in the plants at an early point in development, which continues to affect the plant's food production even after we remove the switch. So it seems that plants already have this layer of regulation, and all we did is tap into it. So the next step would be to discover how to do it using the plant's existing genetics." Unquote. Alright, so let me summarize what's happening here. The researchers essentially identified a genetic regulatory switch, and when you turn it off with this FTO protein, it leads to upregulated plant growth, leading to bigger root systems, which means they can absorb more water, and this confers greater drought resistance. And because that can contribute nutrients and water to the shoots, this above-ground tissue, including the leaves, stems, flowers, everything else, this leads to not just boosted yields, but even improved photosynthesis. This is amazing. Most research that attempts to boost yields is only able to do so in small increments, think like 5 to 15% maybe. But this is huge. A 50% increase in yields is groundbreaking, no pun intended. And the implications here are world-changing, and I mean that literally. If this technology is truly viable and it can be applied at scale, it could be an extremely potent tool in our fight against climate change. To quote Dr. He again, he said, quote, "Early field studies show it can be scaled up. We hope to work with academia and industry to further understand this biology and to safely and widely apply this new technology. Even beyond food, there are other consequences of climate change. Perhaps we could engineer grasses in threatened areas that can withstand drought. Perhaps we could teach a tree in the Midwest to grow longer roots so that it's less likely to be toppled during strong storms. There are so many applications." Unquote. There really are a ton of applications. Just off the top of my head, I'm also imagining deeper, stronger, more extensive root systems would be much better at holding the soil together, so we might have fewer issues with runoff or maybe even landslides, depending on what area of the world we're talking about. This is the kind of science that we need right now. This is the kind of stuff that can really change the world for the better.